Hey guys, as promised, I am recording this supplemental lecture as an addition to our coverage of Chapter 2 on prenatal development and genetics. In this particular lecture, I will be focusing on the topic of genetics with a little bit of review of some concepts we've already discussed. As we have discussed in previous lectures, a single cell is created when a male reproductive cell, a sperm, pushes through the membrane of the ovum, the female reproductive cell. These gametes, as the male and female reproductive cells are also known, each contain huge amounts of genetic information. About an hour or so after the sperm enters the ovum, the two gametes suddenly fuse, becoming one cell, a zygote. The resulting combination of their genetic instructions, over 2 billion chemically coded messages, is sufficient to begin creating a whole person. The blueprints for creating a person are stored and communicated in our genes, the basic units of genetic information. The roughly 20,000 or so human genes are the biological equivalent of software that programs the future development of all parts of the body's hardware. All genes are composed of specific sequences of DNA, deoxyribonucleic acid molecules. The genes are arranged in specific locations and in a specific order along 46 chromosomes, rod-shaped portions of DNA that are organized in 23 pairs. However, sex cells, the ova and sperm, contain half this number, so that a child's mother and father each provides one of the two chromosomes in each of the 23 pairs. The 46 chromosomes in 23 pairs in the new zygote contain the genetic blueprint that will guide cell activity for the rest of the individual's life. Through a process called mitosis, which accounts for the replication of most types of cells, nearly all the cells of the body will contain the same 46 chromosomes as the zygote. Specific genes and precise locations on the chain of chromosomes determine the nature and function of every cell in the body. For instance, genes determine which cells will ultimately become part of the heart and which will become part of the muscles of the leg. Genes also establish how different parts of the body will function, how rapidly the heart will beat, or how much strength a muscle will have. As I stated, humans have 46 chromosomes on 23 pairs. The first 22 pairs are referred to as the autosomes, while the 23rd pair contains the sex chromosomes. One member of each pair is inherited from each parent. If each parent provides just 23 chromosomes, where does the potential for the vast diversity of human beings come from? The answer resides primarily in the nature of the processes that underlie the cell division of the gametes. When gametes, sperm and ova, are formed in the adult human body in a process called meiosis, each gamete receives one of the two chromosomes that make up each of the 23 pairs. Because for each of the 23 pairs, it is largely a matter of chance which member of the pair is contributed, there are 2 to the 23rd power, or some 8 million different combinations possible. Furthermore, other processes, such as random transformations of particular genes, add to the variability of the genetic brew. The outcome, tens of trillions of possible genetic combinations. With so many possible genetic mixtures provided by heredity, there is no likelihood that someday you'll bump into a genetic duplicate of yourself, with one exception, an identical twin. Although it doesn't seem surprising when dogs and cats give birth to several offspring at one time, in humans, multiple births are cause for comment. They should be. Less than 3% of all pregnancies produce twins, and the odds are even slimmer for 3 or more children. Why do multiple births occur? Sometimes a cluster of cells in the ovum splits off within the first 2 weeks after fertilization, during the germinal period. The result is two genetically identical zygotes, which, because they come from the same original zygote, are called monozygotic. Monozygotic twins are twins who are genetically identical. Any differences in their future development can be attributed only to environmental factors, since genetically they are exactly the same. There is a second and actually more common mechanism that produces multiple births. In these cases, two separate ova are fertilized by two separate sperm at roughly the same time. Twins produced in this fashion are known as dizygotic twins. Because they are the result of two separate ovum, sperm combinations, they are no more genetically similar than two siblings born at different times. Of course, not all multiple births produce only two babies. Triplets, quadruplets, and even more births are produced by either or both of the mechanisms that yield twins. Thus, triplets may be some combination of monozygotic, dizygotic, or trizygotic. Although the chances of having a multiple birth are typically slim, the odds rise considerably when couples use fertility drugs to improve the probability they will conceive a child. For example, 1 in 10 couples using fertility drugs have dizygotic twins, compared to an overall figure of 1 in 86 for all Caucasian couples in the US. Older women, too, are more likely to have multiple births, and multiple births are also more common in some families than in others. The increased use of fertility drugs and rising average age of mothers giving birth has meant that multiple births have increased in the last 25 years. Boy or girl, establishing the sex of the child. Recall that there are 23 matched pairs of chromosomes. In 22 of these pairs, each chromosome is similar to the other member of its pair. The one exception is the pair, which is the one that determines the sex of the child. In females, the 23rd pair consists of two matching, relatively large X-shaped chromosomes, appropriately identified as XX. In males, on the other hand, the members of the pair are dissimilar. One consists of an X-shaped chromosome, but the other is a shorter, smaller Y-shaped chromosome. This pair is identified as XY. Each gamete carries one chromosome from each of the parent's 23 pairs of chromosomes. Since a female's 23rd pair of chromosomes are both X's, an ovum will always carry an X chromosome, no matter which chromosome on the 23rd pair it gets. A male's 23rd pair is XY, so each sperm could carry either an X or a Y chromosome. If the sperm contributes an X chromosome when it meets an ovum, which remember will always contribute an X chromosome, the child will have an XX pairing on the 23rd chromosome pair and will be a female. If the sperm contributes a Y chromosome, the result will be an XY pairing, a male. It is clear from this process that the father's sperm determines the sex of the child. This fact is leading to the development of techniques that will allow parents to increase the chances of specifying the sex of their child. In one new technique, lasers measure the DNA in sperm. By discarding sperm that harbor the unwanted sex chromosome, the chances of having a child of the desired sex increase dramatically. Of course, procedures for choosing a child's sex raise ethical and practical issues. For example, in cultures that value one sex over the other, might there be kind of a sex discrimination prior to birth? Furthermore, a shortage of children of the less preferred sex might ultimately emerge. Many questions remain then before sex selection becomes routine. What determined the color of your hair? Why are you tall or short? What made you susceptible to hay fever? And why do you have so many freckles? To answer these questions, we need to consider the basic mechanisms involved in the way that genes we inherit from our parents transmit information. We can start by examining the discoveries of an Austrian monk, Gregor Mendel, in the mid-1800s. In a series of simple yet convincing experiments, Mendel cross-pollinated pea plants that always produced yellow seeds with pea plants that always produced green seeds. The result was not, as one might guess, a plant with a combination of yellow and green seeds. Instead, all of the resulting plants had yellow seeds. At first, it appeared that green-seeded plants had no influence. However, Additional research on Mendel's part proved that this was not true. He bred together plants from the new yellow-seeded generation that had resulted from his original crossbreeding of the green-seeded and yellow-seeded plants. The consistent result was a ratio of 3/4 yellow seeds to 1/4 green seeds. Why did this 2-to-1 ratio of yellow to green seeds appear so consistently? It was Mendel's genius to provide an answer. Based on his experiments with pea plants, he argued that when two competing traits, such as a green or yellow coloring of seeds, were both present, only one could be expressed. The one that was expressed was called a dominant trait. Meanwhile, the other trait remained present in the organism, but unexpressed. This was called the recessive trait. In the case of Mendel's original pea plants, the offspring plants received genetic information from both the green-seeded and yellow-seeded parents. However, the yellow trait was dominant, and consequently, the recessive green trait did not assert itself. Keep in mind, however, that genetic material relating to both parent plants is present in the offspring, even though it cannot be seen. The genetic information is known as the organism's genotype. A genotype is the underlying combination of genetic material present but outwardly invisible in an organism. In contrast, a phenotype is the observable trait, the trait that is actually seen. Although the offspring of the yellow-seeded and green-seeded pea plants all have yellow seeds, the genotype consists of genetic information relating to both parents. And what is the nature of the information in the genotype? To answer that question, let's turn from peas to people. In fact, the principles are the same, not just for plants and humans, but for the majority of species. Recall that parents transmit genetic information to their offspring via the chromosomes they contribute through the gamete they provide during fertilization. Some of the genes from pairs, called alleles, genes covering traits that make alternate forms, such as hair or eye color. For example, brown eye color is a dominant trait. Blue eyes are a recessive trait. A child's allele may contain similar or dissimilar genes from each parent. If the child receives similar genes, he or she is said to be homozygous for the trait. On the other hand, if the child receives different forms of the gene from its parents, he or she is said to be heterozygous. In the case of heterozygous alleles, the dominant characteristic, brown eyes, is expressed. However, if the child happens to receive a recessive allele from each of its parents, and therefore lacks a dominant characteristic, it will display the recessive characteristics such as blue eyes. In reality, relatively few traits are governed by a single pair of genes. Instead, most traits are the result of polygenic inheritance, through which a combination of multiple gene pairs is responsible for the production of a particular trait. Furthermore, some genes come in several alternate forms, and still others act to modify the way that particular gene traits produced by other alleles are displayed. Genes also vary in terms of their reaction range, the potential degree of variability, and the actual expression of a trait due to environmental conditions. And some traits, such as blood type, are produced by genes which neither member of a pair of genes can be classified as purely dominant or recessive. Instead, the trait is expressed in terms of a combination of the two genes. Sometimes the dominant gene does not completely suppress the recessive gene. This is called incomplete dominance. An example of this can be found in the recessive gene disorder sickle cell disease. The gene that produces healthy round-shaped red blood cells is dominant. The recessive gene causes an abnormality in the shape of red blood cells. They take on a sickle form, which can clog the veins and deprive vital organs of oxygen and increase the risk of stroke. To inherit the disorder, a person must receive the recessive gene from both parents. Those who have inherited only one recessive gene are called carriers and should be unaffected by this recessive trait. Yet, Carriers of sickle cell have some red blood cells that take on the C-shaped sickle pattern. Under circumstances of oxygen deprivation, such as high altitudes or physical exertion, carriers for the sickle cell gene may experience some of the symptoms of sickle cell. Most of the known genetic disorders are dominant gene linked. However, the vast majority of dominant gene linked disorders are not serious or debilitating. For example, the majority of those with Tourette syndrome suffer only minor tics from time to time and can easily control their symptoms. Huntington's disease is a dominant gene linked disorder that affects the nervous system and is fatal, but does not appear until midlife. Recessive gene disorders, such as cystic fibrosis and sickle cell anemia, are less common but may actually claim more lives because they are less likely to be detected, as people are unaware that they are carriers of the disease. Some genetic disorders are sex-linked, meaning that the defective gene is found on the X chromosome. Males only have one X chromosome, so are at a greater risk for sex-linked disorders due to a recessive gene, such as hemophilia, color blindness, and baldness. For females to be affected by the genetic defects, they need to inherit the recessive gene on both X chromosomes. But if the defective gene is dominant, females can be equally at risk. Table 2.1 lists several genetic disorders. A chromosomal abnormality occurs when a child inherits too many or too few chromosomes. The most common cause of chromosomal abnormalities is the age of the mother. As the mother ages, the ovum is more likely to suffer abnormalities due to longer-term exposure to environmental factors. Consequently, some gametes do not divide evenly when they are forming. Therefore, some cells have more than 46 chromosomes. In fact, it is believed that close to half of all zygotes have an odd number of chromosomes. Most of these zygotes fail to develop and are spontaneously aborted by the mother's body. One of the most common chromosomal abnormalities is on pair 21. Trisomy 21, or Down syndrome, occurs when there are 3 rather than 2 21st chromosomes. A person with Down syndrome typically exhibits an intellectual disability and possesses certain physical features, such as short fingers and toes, folds of skin over the eyes, and a protruding tongue. There is as much variation in people with Down syndrome as in most populations, and those differences need to be recognized and appreciated. Other less common chromosomal abnormalities of live-born infants occur on chromosome 13 and chromosome 18. Refer to Table 2.2 on the prevalence of these chromosomal disorders in our home state of Illinois, or in the author's home state of Illinois. When the abnormality is on the 23rd pair, the result is a sex-linked or chromosomal abnormality. A person might have XXY, XYY, XXX, or XO. Two of the more common sex-linked chromosomal disorders are Turner syndrome and Klinefelter syndrome. Turner syndrome occurs when part or all of the X chromosomes is lost, and the resulting zygote has an XO composition. This occurs in one of every 2,500 live female births, and affects the individual's cognitive functioning and sexual maturation. The external genitalia appear normal, but breasts and ovaries do not fully develop, and the woman does not menstruate. Turner syndrome also results in short stature and other physical characteristics. Klinefelter syndrome, XXY, results when an extra X chromosome is present in the cells of a male and occurs in about 1 of 700 live male births. The Y chromosome stimulates the growth of male genitalia, but the additional X chromosome inhibits this development. An individual with Klinefelter syndrome has some breast development, infertility, and has low levels of testosterone. Behavioral genetics is the scientific study of the interplay between the genetic and environmental contributions to behavior. Often referred to as the nature-nurture debate, Gottlieb, 1998, 2000 and 2002 suggest an analytic framework for this debate that recognizes the interplay between the environment, behavior, and genetic expression. This bidirectional interplay suggests that the environment can affect the expression of genes, just as genetic predispositions can impact a person's potentials. Additionally, environmental circumstances can trigger symptoms of a genetic disorder. For example, a person who has sickle cell anemia, a recessive gene linked disorder, can experience a sickle cell crisis under conditions of oxygen deprivation. Someone predisposed genetically for type 2 diabetes can trigger the disease through poor diet and little exercise. Research has shown how the environment and genotype interact in several ways. Genotype environment correlations refer to the processes by which genetic factors contribute to variations in the environment. There are three types of genotype environment correlations. Passive genotype environment correlations occur when children passively inherit the genes and environments their family provides. Certain behavioral characteristics, such as being athletically inclined, may run in families. The children may have inherited both the genes that would enable success at these activities and given the environmental encouragement to engage in these actions. Figure 2.3 highlights this correlation by demonstrating how a family passes on water skiing skills through both genetics and environmental opportunities. Evocative genotype environment correlations refer to how the social environment reacts to individuals based on their inherited characteristics. For example, whether one has a more outgoing or shy temperament will affect how he or she is treated by others. Active genotype environment correlations occur when individuals seek out environments that support their genetic tendencies. This is also referred to as niche picking. For example, children who are musically inclined seek out music instruction and opportunities that facilitate their natural music ability. Conversely, genotype environment interactions involve genetic susceptibility to the environment. Adoption studies provide evidence for genotype environment interactions. For example, the Early Growth and Development Study followed 360 adopted children and their adopted and biological parents in a longitudinal study. Results have shown that children whose biological parents exhibited psychopathology exhibited significantly fewer behavior problems when their adoptive parents used more structured parenting than unstructured. Additionally, elevated psychopathology in adoptive parents increased the risk for children's development of behavior problems, but only when the biological parent's psychopathology was high. Consequently, the results show how environmental effects on behavior differ based on genotype, especially stressful environments on genetically at-risk children. Epigenetics studies modifications in DNA that affect gene expression and are passed on when cells divide. Environmental factors such as nutrition, stress, and teratogens are thought to change gene expression by switching genes on and off. These gene changes can then be inherited by daughter cells. This would explain why monozygotic or identical twins may increasingly differ in gene expression with age. For example, Fraga et al. 2005 found that when examining differences in DNA, a group of monozygotic twins were indistinguishable during the early years. However, when the twins were older, there were significant discrepancies in their gene expression, most likely due to different experiences. These differences included susceptibilities to disease and a range of personal characteristics. The field of behavioral genetics and the emerging field of epigenetics have done a great job of showing us the degree to which nature and nurture interact in an infinitely complex way. Genetics will nudge us in a certain direction. However, the role of environment cannot be denied in this process.